Hi guys, welcome to another homework video episode. Today we are learning about the central dogma. So please update your table of contents with the central dogma homework notes and we will get started. So the central dogma of biology. Does anyone know what that means? Please answer for yourself. So a dogma is actually a belief that we know to be absolutely true. So some of you might have your own dogmas, things that you really believe are true. But the central dogma of biology has to do with DNA, which might not surprise you. So the central dogma of biology is the fact that DNA, right here, replicates and then is then transcribed into RNA and then is translated into proteins, and then those proteins make us. So this is the belief that we know to be absolutely true in bio, is that DNA creates RNA, which creates proteins, which basically means that DNA creates everything about us, right? It's the starting point for everything that makes up organisms. So I want you to actually write all of this down. You should know what a dogma is, and then you should know the central dogma of biology is actually this process. So you can write it just like this, with the arrows, with the words, all of it. All right, so the first step is replication, and it's when we make a copy of the DNA. So it's from DNA to DNA, and again, you want to write all this down. There's an enzyme that's called a helicase that unwinds the DNA. Remember, DNA is in the shape of a helix, so helicase unwinds the helix. Then another enzyme, DNA polymerize, is to make. So DNA polymerase is the one that makes DNA, basically. And it adds bases A, T, C, and G. All of this occurs in the nucleus of a cell. So this is the replication phase of the central dogma. We make more DNA by unwinding the helix and adding new bases. You don't have to write this, but this is just a visual. So here's a strand of DNA. We separate the DNA using helicase, and then an enzyme called DNA polymerase will come in and add the new bases. Remember, A pairs with T and C pairs with G. And then in the end, we have two new strands that are identical to each other, but they are half new and half old, which is called semi-conservative. Another visual of it, so we open it using helicase. This is called the replication fork. Kind of looks like a fork if you look at it. Then there's another enzyme actually called primase that will lay down a little bit of primer. That's how the body knows where to start making a new DNA strand. Then DNA polymerase will come in, find the primer, and start laying down nucleotides, or the little bases. It happens in the 5' to 3' direction, so they actually go in opposite directions. And after you add all the new bases, we end up with two new strands that are the exact same, remember, because we made a copy. Good, so step 1 is complete. Excellent. On to transcription and translation. So transcription is when we use DNA to make RNA, specifically mRNA, which is written as tiny m, big RNA. So this is our blue DNA down here, and this big blob is the enzyme RNA polymerase, and it is going to lay down nucleotides to create an RNA template, which is this red coming out here. Recognize that RNA uses A, U, C, and G, so that U replaced the T that was in the DNA. This also occurs in the nucleus, but then the mRNA that is created is then sent out of the nucleus and into the cytoplasm of the cell. So we're in the transcription phase, going from DNA to RNA. So if you were to transcribe this DNA molecule, first we would replicate it. So it needs its buddy, right? A, T, A, T, A, T, A, T, C, G, C, G, right? But then you take this strand and you turn it into RNA, which is A, U, C, and G. So it would look like this. So T now pairs with AT, but A, instead of pairing with T, has to be a U. So anything that would have been a T is now a U because we are in RNA. So now we've done replication and transcription. All that we have left is translation. So again, DNA to DNA is replication, A, T, C, and G. Transcription is DNA to RNA, A, U, C, and G. And then this is translation. Looks a little bit complicated, but basically you take that red RNA that we saw, and there's the ribosome. Remember the ribosomes in the cell that make proteins? Well, the ribosomes bring in these little tRNAs, and they read the mRNA to bring in amino acids. So if you were to look at it like this, here's the mRNA down the bottom. This big double blob is the ribosome. And there are tRNAs that match up with the mRNA and have amino acids attached. And so they just come down the line and they keep adding amino acids. And this protein will get bigger and bigger and bigger until it's released into the cell. You do want to write down that ribosomes themselves are actually also made of RNA. It's just called rRNA, which is known as ribosomal RNA. You'll have to know that for class. So translation is our last step. It's when the ribosome reads the mRNA and translates it into tRNA. So we're still in RNA, so we still have A, U, C, and G. No T's. It's the process of going from RNA to protein. The ribosome reads the RNA 3 letters at a time. So if you look down here, they're in little groups of 3. That's called a codon. Those groups of 3s are called codons. And for every codon, it adds the correct amino acid until eventually you get a protein. So this is the process of going from that RNA strand to a protein. If we look at this zoomed in, here's our ribosome, here's our little triplet code called codons, and you can actually use a special chart that we know that the body uses, not a real chart, but the body just knows, that every 3 bases codes for a certain amino acid. So you can use this chart here. So say the first letter is an A, you'd go here. Say the second letter was a C, you'd be in this box. And say the third letter, which is over here, was a C also. ACC codes for THR, which stands for threonine, which is an amino acid. So if we look at this down here, GCA codes for, this says alanine, let's see, GCA. GCA is alanine. Perfect. So you'll be doing that in class. One more thing to note is that the 3 letters AUG tell the body to start making a protein. It's known as the start codon. I remember it because I'm born in August, so it's the start of my life. You can remember it that way if you want. And there are 3 stop codons, which tell the body to stop making the protein. They are UAA, UAG, or UGA. UA, UAG, or UGA. So whenever the ribosome reaches one of these, the protein stops and is released. So that's the central dogma, going from DNA, transcribing through RNA polymerase into RNA, and then using the ribosome to create a protein. Yay! Good job.